So the focus of this presentation is on how we're compiling a biological database on deep sea benthic communities around the Antarctic continent. I'll also briefly touch on how this data can be used to answer fundamental ecological questions, such as whether habitat forms facilitate benthic communities. So I'm fortunate to be working on a project that's looking at the patterns and drivers of benthic biodiversity around the Antarctic continent, using existing seafloor images from past voyages, which are shown in this map with the coloured dots. We're aiming to provide the most comprehensive and large-scale investigation using a consistent and transparent and collaborative approach. We're also making all of our data and these images available to everyone. So we're collecting images from all over the world, from far and wide. We're subsetting these images, and then we're extracting biological data. In parallel to this, we're collecting environmental data, and together the biological data and the environmental data will be used to map the distribution of biota around Antarctica. For this presentation, I'm going to be focusing on how we reach the destination highlighted in the red box. But if you want to hear more about aims 2 and 3, so the environmental data and the outputs that we're hoping to produce, I encourage you all to see the material that's uploaded by Jan and Nicole. So as a starting point, we're looking at all benthic images that meet these criteria. So we want images that are from downward facing cameras. We want the location of those images to be known, and we want to be able to make estimates of size of those images. So the red dots on the left-hand image here shows the locations of laser points, which can be used to calculate both the size of the image and objects within those images. We then, from this 60,000 plus set of images, we then subset these images to ensure that we get images that are scoreable so that we can extract biological data and that our samples are spatially balanced so that we're not sampling some areas more intensely than others. To do this, we're using an MBH sampling design and selecting images at approximately every 100 metres along transect lines. This approach is really fantastic because it's also expandable, so we can increase our sampling effort over time without increasing or introducing any spatial biases. So after selecting the images that we're going to be analysing, so the subset of images, we're then using 2 programs to extract the biological data. The first program is CoralNet, which we use to get percentage cover calculations for the different biota. So this program just assigns 108 evenly spaced points, and we just tell it what's located at each of those different points. The second program we're using is Beagle, which is used to get count data for mobile species. So to get this data, we systematically go through each of our images and we search for all these often obscured mobile critters, and we draw circles around them. And not only does this give us count data, but the size of the circles also gives us size estimates for the different biota that occur. So size is obviously really important. We can't just assume that all brittle stars, for example, are the same size. So here the 2 red circles show very differently sized brittle stars. The larger one is in fact a massive brittle star, and the smaller ones are more standard sized brittle star. And to provide a bit of scale here, the green circle shows the size of an octopus. So we're annotating our images based on the Catani classification scheme. And this scheme is based on relatively easy to recognise morphotaxa, which can indicate the functional role of those taxa. It's a useful approach because it's often very difficult to identify finer scale species IDs, which can be limited by the quality of the images, for example, or the fact that some animals might be obscured by something in the image. So after classifying all the points and all of the biota in the images, Beagle and CoralNet help produce these image libraries which show cropped version of every single point or every single animal that we annotate and how it's being classified. This is obviously really useful for our own verification purposes. So every annotation we at least double check to ensure consistency and accuracy, but it also helps with transparency. So if people have different views of our classification, they're free to look over it and challenge any discrepancies. It also allows us to get targeted help from people with particular taxonomic expertise. For example, if we were struggling with some of the IDs of our Psidians, we could just send off folders of images of Psidians for them to provide feedback. So the other thing that these libraries are currently being used for is to provide training material to assist in automated object recognition and detection with machine learning, which will be further developed to speed up future analysis of images. So hopefully in the future, we don't have to sit down and manually annotate images. Many of the organisms can be identified automatically, and it's just a matter of verification. So far, our image libraries and our biological database are based on approximately 250,000 points in CoralNet and over 100,000 different mobile taxa identified in Beagle. But this is growing. So this biological data in combination with the parallel environmental data will then feed into the production of species distribution maps, which is a main goal of this research. But potentially there are some other uses of these databases. For example, it can be used to answer some ecological questions. So we know that environmental factors are strong drivers of species distribution for these benthic assemblages, but we don't really know the extent to which species interactions shape these communities. This is certainly true for opportunistic species such as brittle stars, which appear to be distributed everywhere. And we don't know if the nature of these relationships depends on habitat type. So to understand this in a bit more detail, we need to be able to separate co-occurrence that's due to shared environmental preferences from potential species interactions. There are a number of different approaches that you can use to do this, but here we're looking at approaches that can accommodate abundance or biomass data rather than just presence and absence data. So for example, we can use this method demonstrated by Hui et al., which uses a model-based approach and looks at latent variables to try and get proxies for potential species interactions. So it separates those environmental correlations from potential species interactions. You can then look at whether the nature of these interactions, so the amount of positive interactions and negative interactions changes with these different habitat types. And this is something I'm looking at doing. And you can sort of test some sort of fundamental ecological theories. For example, the stress gradient hypothesis, which says that under more stressful environments, positive species interactions are likely to be more prevalent. So I haven't started on this analysis yet, but there are certainly some good signs that there might be different sorts of relationships for these different habitat types. So here we've just got a plot of the abundance of brittle stars in our benthic images against the percentage cover of habitat forming species, all habitat forming species. And there seems to be this suggestion that the nature of this relationship is actually different for the different habitat types. So for example, steep and flat benthos seem to have different relationships. I hope to be able to share more on this a bit soon in a short while. So the point I want to end on, I guess, is that this project is all about collaboration. We're relying on researchers from across the world to contribute their images, and we would encourage anyone who has images that they think might be useful to get in touch. I also want to make a shout out to anyone with any taxonomic expertise to get in touch and help with the fine tuning of our image libraries and contribute in that way. So there's some obvious advantages with working collaboratively. We're able to collect more data. We can prioritise future data collection, image collection, where we conduct voyages, that kind of thing. We can harness expertise which might be distributed widely across the globe. And I think the development of some standard operating procedures around this would be very helpful. So standard operating procedures around image collection and the annotations would be fantastic and help this process. So yeah, I'd just like to encourage you all to get in touch if you'd like to be involved. Finally, I want to thank my research institute, the ARC for funding this project, and all of our different collaborators that we have. Thanks.